Please all participants download this simplified corrected file and save in working directory. Okay, please first copy the name of these two files. Paste here. Dot txt
Okay,
corrected
sir.
corrected dot txt and second file name is
Which file, sir?
second file.
Sample info.
No, this is for Drosophila. I think it's for mouse.
One small counts and one was sample
1 minute,
info, I
please.
think.
Sample file info. Yes, small count dot text. Small,
This one.
yes, small underscore count dot text. Paste here. Dot txt.
Yes.
Okay. Now we first we need these files in screen number 3. It's mean we want to import this file in R. So copy the name of these two files. Provide to ChatGPT. Paste here. And write a message. I want to import these files in R from working directory.
Okay, sir.
सेंड करें मैसेज.
Yes.
स्क्रिप्ट को कॉपी करें. और उसको रन करें. इसके लिए आप फंक्शन यूज करेंगे रीड डॉट टेबल. सेकंड भी यूज करें. ओके, अब दोनों फाइलें आपके पास वर्किंग डायरेक्टरी में मौजूद है. ये रन करने की जरूरत नहीं है. ये वाले कोड ना, ये
Yes, sir.
वर्किंग डायरेक्टरी के लिए.
This one. Okay, sir.
ये कोड आपने रन नहीं करना है, ठीक है. अच्छा, अब हमारे पास फाइल्स मौजूद हैं.
Yes, sir.
अब हमने इसका एसटीआर करना दोनों फाइल्स का. रन करें इसको. ओके, दूसरी फाइल का भी एसटीआर करें.
Okay, sir.
ओके, sorry, some participants are unable to understand Urdu. Okay, now please run copy these two strs from screen number 2. Copy, open ChatGPT. Open new chat. Okay. Now write a message. I want to perform RNA-seq analysis. Please write script step by step for all analysis. Please write script step by step for all analysis. Okay, first copy, first code, install and load required libraries. I think you have already all these packages. You just need to do libraries. Please run libraries. Okay, all libraries are working. Now copy second step, prepare the data. You already have, please show me script from ChatGPT. It is showing you already have sample info and small count. The next step is to ensure that the count matrix is in the correct format, and then we can proceed to create the ASIC2 dataset. Okay, to run these lines to confirm matrix is correct or not correct. Next, but it is showing you length of dimensions not equal to array extent. You can check in screen number 3. Variables and observations should be same. So again, run next code. It is again showing you an error. Row name do not match between the count matrix and sample information. So row name, count matrix, and sample info. In count matrix, you have 10 observations, while in variable you have 4, and 4 variables and 12 observations. So copy these 2 errors and provide to ChatGPT. Just errors, just error message, yes, from here. Send this message. I think ChatGPT is working. No. I click on refresh button and paste in previous. Okay. So it is showing you the first read instruction. The error message you are seeing suggests that the length of row names count matrix does not match the length of sample info, or that the length are not compatible for comparisons. Here's how to debug this step by step. First, check the length of row names and check the extra or missing rows. Okay, first run first code, second code, and provide the result of these first and second code to ChatGPT. Check length, 10th, 12th. No, check for extra or missing rows. It is showing you all rows sample info are false. In row names, longer object length is not a multiple of shorter object length. It's mean false. Here should be true, but your answer is false. So check ChatGPT. There may be another code. Scroll down. Yes. No, run this line. It is showing you 12 lines. Okay. It should be 10 or 12 for both. Okay, now copy the results with warning messages from length. First, also include above line. Yes. No, 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 not error. Just for length row names. Copy, paste here, and run. First read instruction in first line. It looked like the mismatch between the row names and sample info is due to the fact that their length are different, 10 rows and 12 rows. This is causing the error because you are trying to compare two vectors of different length. That's why you are getting errors again and again. And R cannot perform elements-wise comparison if the length are not the same. So here how you can address these issues. Check which rows of count matrix and sample info should match. You should have 10 rows in count matrix but 12 in sample info sample names. You might need to check if the sample info files contain extra or missing entries. Maybe you have missing entries like 10 rows or maybe extra rows. So you need to check which one are extra or which ones are missing. So check if there are extra entries in the sample. Run this code. And run this line. Okay. Now copy next code. If there are extra row in sample info. Copy and run. Run this line. It will show you extra row. Okay, for this you need to click on sample info. Just select sample info and run. Now you have 0 observations. Okay, import first file again, sample info. Line number 5. Yes. Click on line number 5. Uh, sorry, screen number 3, open sample info file. Here are 12 observations and a sample count file. Here is only 4 sample, 10 observations. In sample count, the file name, variable name should be not sample 1, sample 2, sample 3. Sample name should be, click on sample info. Sample name should be MCL1 DG, MCL1 DH, MCL1 DH. So this file is not correct. I think you are using a small info file, not corrected file. Your file name is sample info. Check script. Yes, sample info corrected. And also import sample info file and compare both these files. Copy the script, copy line number 5 and paste in line number 6. And change its name correctly to sample info .txt and run this line. And first change its name sample info from yes, sample info 1. Okay, run this line. Sample info 1
Yeah.
have 12 observations, 4 variables. Same file.
Yes, sir.
Corrected is also same. Wait, please. Sorry, this file is the data set in this file is not corrected. If you open your previous file, click on 68 or 85 file, zip file.
This
Yes,
one.
this one. And check both files. Not this one. For Drosophila file.
88.
Yes, for Drosophila.
It is 88.
Yes.
Yeah.
Count and you can check the headings are primers names or markers name SSR. Close and check row names from sample info.
Sorry, sir.
Close this file and check sample info Drosophila. Yes, you can check sample name and row names are same in both files of Drosophila. But in the case of mouse, you can check their variable names and here are rows names are same. But in the case of mouse data, these files are not accurate. You can compare it with mouse file. Open mouse file and check both variable name and row names are different. You can check sample name and sample info. In sample info, these are
Camel info,
and small count.
small count, sorry.
Yes, you can check here. Here is sample 1, sample 2, sample 3, 4 instead of row names.
So row names should be same.
Row name and variable
Order.
name should be same.
So what about here? This one,
Wait, please. Open. Yes, open this one. Here you can see variables name. These are variable name.
which one are the variable name?
These are variable name.
FB. Okay.
And these names, if you click on small count, sorry, sample info,
Hmm.
click on sample info. You
Yeah.
can check these variable names and these rows names are same.
These are column. Okay.
These variable names, these rows names and variables name
Hmm.
in count Drosophila data are same.
Hmm.
That's why these files is correct. But in mouse data, if you click on here are the sample names.
These.
These are sample names.
Oh.
But
Oh.
small count, this is not correct. Here are sample 1, sample 2, sample 3, sample 4. So this data set is not correct data set. Maybe this file, I think I have copied for another data set, another organisms. That's why you are facing again errors again and again.
Sir, I I ran two plot there successfully plotted, but the
You
heat
have
map there
created
having
just
issue.
only count data file or only just for this
Maybe.
one.
I
When
think
you
so.
combine both, then it will give you error
Okay, I
for
see.
DEA-seq
I think the
analysis.
error the error is between the 12 and 10. The is
Not
the row
not
and the one in the row matching.
between 10 and 12, between variable names. Here
Okay.
variable names are sample 1, sample 2, sample 3, but observations names are genes name are markers names.
And
Reference
even
is
sometimes the vector is also not matching.
you need to remove not mismatched characters. Okay.
Sir, I
Now,
have a question.
yes.
Uh, Sir, all these information can be in one file or no? These should be in two files for making
Yes, it
graphs.
can be in one file also. Depend
Hmm.
on companies. They maybe give you an Excel file.
Yeah, in one file.
Yes, no issue. If you
Hmm.
have in one file,
So can
you
we
can
do
use.
practice in that one file if
Yes,
you
you
have?
can use. Okay. Yes, you can
Uh,
do practice. Okay, open ChatGPT and write a message. Open new chat.
yes.
Open new chat. I want to do practice for RNA-seq analysis in R. from where I get count, data and sample info. Sample info .txt file, question mark and send message. So here are some sources from where you can get these files. First is NCBI, GEO, Gene Expression Omnibus. And GEO is a repository for high throughput gene expression data, including RNA-seq. You can download raw count data along with sample metadata in the format you need, txt.csv. So go to GEO. Here are the steps for finding the data set for RNA-seq for human or other organisms. Click on GEO. Yes, link. It will take you in website GEO and search here and find second step ChatGPT. Follow second step. No, click on ChatGPT. Yes. Second step, search RNA-seq data, such as RNA-seq human, or choose a data set based on your species of interest. Download the relevant data set. You will typically find count data in the series matrix files or along with sample metadata in associated files. Okay, click on NCBI website and search here in search bar RNA-seq mouse, or just type mouse and search. There are results for mouse. Two results are found. You can click anyone. Just click on this. So here are data set in the form of txt. Its description is also given here. Its complete detail. Here you can download this file in txt file format. But first read about this data set, or detailed about this data set. And the liver play essential role in the maintaining systemic glycolipids hemostasis under ever-changing metabolic stress. I think this data set consists of mouse liver data. Okay, now click on download data txt. So here are a series of sock files, GZ and series matrix txt.gz and you can download data set according to your requirement. Minimal file series matrix files, open matrix files. Click on matrix file. Download this file and open this file.
Mhm.
It is close this file This file is in zip folder, open folder.
This is
You need
.dz.
to extract this file. This is the sample info file. Okay, check another files.
Which ones?
In supplementary data, you can find gene count text. Second last, yes. This is for gene count. This data set is for gene counts. So we need two files with sample info and count data. We have we have a count data. We need to find sample info. Okay, close it and find sample info file. And try series soft file. First one.
This is, sir, which file?
Extract this file.
Already extracted.
Open.
Yeah. Yes.
Scroll down. I think this is supplementary file. Sample title. Okay, import count file in R. We have download gene count txt file. So please import this file in R. Run first line. Okay, click on this file. Okay. Now take STR. Copy this STR. This file is not a raw file. It is a matrix file. Copy. Open ChatGPT. Open new chat. And write a message. Please explain this file. Send message. It is showing you the file you have shared appear to be a data frame with 3,385 rows, observation, and 20 columns, likely representing gene expression data or genomic information. So this file consists of both information, data, and information. Here is the breakdown of each column in data. Gene ID and okay, scroll down. Please scroll down. Last lines. Summary. This dataset likely contains information on the expression level of genes under various experimental conditions, as well as the basic genomic data such as gene identifiers. Okay. Now say thanks. Now I want to perform RNA-seq analysis. In R. Can I perform RNA-seq on this file? Because now you have all dataset in single file. Can I perform RNA-seq on this file? Question mark and send. Yes, you can perform RNA-seq analysis in R using this file, but it depends on the specific type of RNA-seq analysis you want to do and the structure of your data. Based on your data set, it look like you already have done expression data. The values under column like TWT1 to which might be count or other numeric values related to gene expression. Step perform RNA-seq analysis. So check data format, install necessary packages. Okay, scroll down. Scroll down. Prepare dataset and okay. Now try these steps one by one. You already have these tools. Do library.
Leave.
You already have, I think, Lima. You can remove all these codes.
So
Do library, please, not install. Script for library is given below in ChatGPT.
okay. So
Scroll down. There is no, I think no package. Okay, run export. Next. 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 Okay, next. Copy. It is showing you plot for DDS intergroup conditions. Again, run line number 36. Okay, run line number 44. Unable to find the inherited method for function. Okay, yes. This function is not found in this plot PCA function. So you need to use another method. Try another plot. MA plot. Yes. Okay, next. Volcano plot. It's working. Okay. Now you need to change function for PCA. So copy the error of PCA because this function is not working in plot PCA function. Provide to ChatGPT. Send this message. Okay, first read instruction. It is showing that the error you are seeing is because the plot PCA function from the DE-seq to package expect object, but the error suggests that either the object has not been correctly created or loaded, and there is a version mismatch with the package. To resolve this issue, let's make sure the steps are followed correctly. I will walk you through a few check and fix. You can run these codes one by one and check. Next. Okay, again copy and also copy the above code which we have run from screen number 2. Also show above results. Yes. These ones also include warning message. Okay. Copy and provide to ChatGPT. And other participant also download sample files from NCBI website. So you can download from plant data, animal data, human data by searching name on search bar. Okay, send this message. And try this one by one. Okay. Now, hope you can do after practice. And please also do practice by downloading other files from this website. You can use matrix file. So in matrix file, all data is in single file. So you don't need to combine sample info and count data one by one. Count data and sample info is very raw data. So, and matrix is its advanced form after raw data. So try to get matrix. Okay, good, you have done. Okay, now also try other analysis and also share the file within group. Okay, thank you.
Okay.
Okay. Okay, please do practice. And in our tomorrow class, we will again work on some sample files for practice for all analysis. Okay, thank you. Allah Hafiz.